introductions while people join in. So hello everyone, thank you for joining our UK Multidisciplinary Centre for Neuromorphic Systems and Computing webinar. It's first ever webinar we are doing, and we hope we will be doing more of them. My name is Natalia, I'm manager of the center. I will be introducing our amazing speakers in 3 minutes. But before that, I wanted to give a little bit of introduction to our center. If you never visited us, or if you've never heard about us at another events, or if you've never seen these slides, probably you've seen that, but anyways. So we are UK Multidisciplinary Centre for Neuromorphic Systems and Computing, UK focal point for neuromorphic computing research in the UK. We are a consortium of 7 universities coordinated at Aston University, and we cover a broad spectrum of topics in neuromorphic computing. Starting in the heart is neuroscience, and then we have photonics, electronics, materials, quantum and physics-inspired computing, like lots of things, machine learning and math modeling, many things. And as I've mentioned, in the heart of our activities is brain-inspired neuromorphic computing. That's why we decided that it would be great to have first topic that we cover in our webinars, someone with neuroscience side. That's why today we have here Eric Hill, who is based at Loughborough University and is reader there. And also we have here Jacky, who is lecturer at Aston University. While people still joining, a couple of more slides from me before. So we have many amazing partners. We're working with them, and we are very open to more collaborations and to more partnerships with industry, with academics as well. We do lots of activities like National Roadmap for Neuromorphic Computing, events like today and bigger scale, some other activities, and if you want to join us as an associated member or as an associated partner, you are very welcome to do this. It is a simple form at the website. Please fill it in and then send us your publications or relevant research, and we are happy to work with you. So again, introductions to our first ever NeuroSYNC webinar. Eric Hill and Jacky will be talking today, and floor is yours.
Thank you very much, Natalia. Whilst my slides just load, I think they're now appeared. If everyone can see that okay. So I'm going to be talking today about some of the biological neuroscience side of the work that we are working on in NeuroSYNC, and how this links to some of the work we've done in the past as well. So I'll be covering in my part of the seminar the engineering of neural circuitry to model biological neural networks. And so this is a large multidisciplinary effort, and a recently completed project for some of the data that I'll be showing today, and also links to Jacky's work as well, is from our EU-funded NewChip project, where a number of different people from different research areas have come together to work on this particular topic. So I've probably missed loads of people out on the list of thanks here that were involved in this project, people like Rishona and some of the groups at Technion that have worked together in this particular area. So one of the inspirations and reasons that we're, I guess, interested in work in this area is concerning energy consumption. So at the moment, we already know that machine learning, artificial intelligence uses a huge amount of energy, and that energy use is increasing massively over the last few years. Especially as we have more deep learning machines, large language models being used in different, lots of different softwares now that you see every day. There's some kind of AI asking you questions. Would you like anything clarified? This is using up a large amount of energy. But we know that the human brain works on much less energy compared to a computer. It's often estimated somewhere around 20 watts. while we're at rest, although that's a lot for the human body. The brain uses a lot of energy in terms of the human body, but compared to computing power, this is a lot less. And we know that human brains are capable of much more complicated tasks than current AI machine learning models. So when we think of current brain usage, we can think of the human brain using around 20 watts of energy perhaps, and we have billions of neurons and probably trillions, if not quadrillion, synapses, connections between those neurons within the human brain, carrying out very complicated tasks all the time. And using in the human body, it's about 20% of our energy use, even at rest. And considering the brain is about 2% of our volume, that's quite a lot for the body. But in terms of computers, we can think of our normal desktop computers, for instance, using amount of energy in the hundreds of watts perhaps, and running quite slowly, and some current supercomputers using huge amounts of energy. And compare that to some of the newer neuromorphic type neuromemorist devices that are using much less energy, have a lot larger number of neurons and synapses perhaps, but nowhere near the complexity of the human brain. And so we really need to understand how human neurons are able to carry out such complex calculations perhaps using so little energy. And so there are, I guess, two routes that we can think about the way human brain cells could be used, from the very biological wetware. It's often described using neurons to actually act as computing components themselves, or can we get the inspiration from how we could maybe design new computing paradigms from the way that the human brain works? And we can also start to use this information to understand why, in some cases, the brain's connectivity and the way that we learn changes. So diseases like Alzheimer's disease, for instance, it changes the way we learn, store information, and there's a huge energy deficit in Alzheimer's disease. We don't use energy properly in those disorders. So again, this will give us more information about how particular disorders may develop as well. So we can start to think about using human brain cells as computing units themselves. And if we think of some areas of the brain, such as the cortex, we have approximately about 80% of the excitatory, the cells in that part of the brain are excitatory. Okay, whereas about 20% of those cells are inhibitory, so maybe those cells putting the brakes on some of that activity. And within that, we have astrocytic cells. Astrocytes were often considered the glue that held the brain together, this support network of cells. And we're understanding more and more that astrocytes, and particularly human astrocytes, play a fundamental role in the way that we learn and within memory formation as well. And so these are some of the very important cell types that we have in the brain, particular areas like the cortex, that perhaps important for our higher functions, that we would want to consider using as computing units. And obviously our brains are very complicated. They've developed over a long period of time. Humans are very slow to develop compared to other species. And our brain builds up these huge networks, these very complicated networks over time. And so developmentally, we can think of how these complex structures, these complex networks developed during the brain, and how these help to maintain memories, how they process memories and process information that's coming into those brain cells. And can we start to recapitulate some of these processes in the lab? And for a long time, it's been really difficult to work with human brain cells. And one of the reasons is access to human tissue. If we think that human brain is stored within our skull, it's very well protected, and for good reason. The brain is very soft. It's a bit like mucus, perhaps, as a biomaterial. And we need every part of that brain. And so we don't really get any volunteers offering to give us brain tissue to work with. And often to work with human brain tissue, it's when people have died and donated their brain tissue. And so this has been a very difficult thing to work with, particularly for areas like disease modeling and drug design, that we haven't had access to the tissues that we needed. But we do know that stem cells, and here I've put a picture of some embryonic stem cells, these cells can turn into any cell within our bodies. So the fact that these cells can go from turning to very unspecialized cells, these pluripotent stem cells, to any cell in the body, including brain cells. And as long as in the lab we can understand the processes that drive that differentiation of those stem cells, the development of those brain cells, we can start to build these cell types in the lab. And so for me, it's a bit like cooking. We have different recipes we can put onto the cells at different times to develop them into different types of cells, and for us, particularly different types of brain cells. We also, because we don't work with embryonic stem cells, the use of stem cells in many areas of science can cause ethical issues, particularly the use of embryonic stem cells. We use induced pluripotent stem cells, and these are a type of stem cell that have the same potential to turn into different cell types as embryonic stem cells, but these are derived from skin cells. And so from the early papers from the early 2000s from Yamanaka's lab that developed these techniques to take somatic cells, so these are cells such as your skin cells, and using very distinct reprogramming factors, able to turn these skin cells into induced pluripotent stem cells. And then we can turn those induced pluripotent stem cells those cells into pluripotent stem cells, which you can then turn into neural stem cells. And from those neural stem cells, we can start to make these different neurological cell types. And so here's a picture of some human iPS-derived neural cells. This culture is about 100 days old in this picture. And what you can see here is a triculture of excitatory cells. You have inhibitory cells in magenta, and this large cell in the center, this green cell, this is an astrocyte. Human brains have more astrocytes than other species, more types of astrocytes. They're more processy, they're larger, and so these maybe are essential to some of our functions within humans. And as I mentioned, this is culture is about 100 days old, and we have grown these cells past a year to 2 years to see how they develop over time. And I guess, unfortunately for us, working with some of these cells in the lab, they take a long time to develop. And so we're talking a period of months, 3 to 6 months perhaps, to get quite mature cells. They're about as slow as humans are to develop. If you were working with mouse embryonic stem cells, these would develop much quicker in the lab than human stem cells do. So we can zoom into this picture a little bit more, and you can really start to see the interactions between these cells. There's many synapses in between these different cells, and the astrocytes get engaged within those synaptic areas and helping to really underpin some of that synaptic activity to protect the neurons. They're fundamental in their protection of neurons, and also the feeding of things like glucose and lactate within those brain cells as well. And so they play a very fundamental role in also tuning the behavior of the neuronal cells. So some advantages of using stem cells as maybe computing components is that we can have control over the initial conditions. And as I mentioned there, we had a triculture, and those tricultures are made up of about 120,000 cells, and of those about 80% are excitatory and about 20% are inhibitory, as well as those supporting astrocytic cells. So we can have control over the network composition and also start to try and gain control over the architecture of those cells. And so we have access to the learning of those cells during their formation, but also not just after as a kind of endpoint assay. So hopefully this video will play. So what you can see here, this is a co-culture of cells. This culture is about 50 days old. And what you can see is at the beginning of that video, the cells are spontaneously active in communicating with each other. We have no input into these cells at that point. And then we've added a drug to those cells. And so when we add that drug to those cells, they start to fire and very much in a very seizure-like way. We're trying to induce a seizure in this particular model by using the drug. And in this raster plot here, you can see that all the cells are firing at the same time. And so we can start to control some of their activity with drugs. Okay. So you go from this very spontaneous level of activity to this synchronized activity, and Jacky will talk about some of these concepts as well later on in his talk, and how the complexity of the network, we can try to gain control of some of those features. We also want to be able to stimulate the cells as well as record from the cells. And so what you saw there was fluorescent calcium imaging, where we're using a dye in that particular case to enable every time the cells release calcium as they're active, this molecule will fluoresce. It's great for short-term experiments, but not necessarily long-term experiments. And so if we want to grow these cells over long periods of time and have inputs and be able to measure outputs from the cells, then we may use a system such as these high density CMOS multi-electrode arrays. And this particular multi-electrode array in this image has over 4,000 electrodes, where we can both stimulate the cells and record from the cells. This does generate a lot of data for us that we have to trawl through to look at the spikes, but it means that we can continuously look at the cell's behavior over time. In other ways that we may want to input into our cells, we can use optogenetics, and I'll talk about that in a moment, where we might actually just be able to stimulate the cells with light as well. And so we have these different approaches to provide inputs, but also to measure outputs. So here's a typical recording from one of these multi-electrode arrays. And what you can see just here is where the cells have been stimulated, just down here in this corner. And you can start to see all these pixels that become illuminated. These are showing various activities of the cells. And so we can do very long-term recordings of these cells over a period of weeks and months, so that we can start to see over time how the cultures develop and how perhaps inputs into this system, particularly designs of inputs, can alter the cell's behavior. And in this picture here, you can see the way that the neurons can grow on top of these different electrodes. These typical electrodes from this 3 brain are sunken within this supporting the material, and we have different levels of contact between the neuronal cells. And in one of these typical multi-electrode arrays, there's about 100,000 neurons within that array in an area that's about 3.8 by 3.8 millimeters in size. So that's quite a lot of neurons within that area for us to work with, to stimulate the cells and to measure their activity over time. I'm going to leave Jacky to explain and talk about this a little bit more, but we can think about how we might stimulate these cells using different patterns. So we can choose different areas of the multi-electrode array and stimulate the cells in a particular pattern and try and link that activity together across the network. And so we can look at how we stimulate those cells and how much energy theoretically that those neurons may use during that training period and afterwards, and how successful different training protocols have been with these particular cells. But I'll leave that to Jacky to explain in more detail. If you look really closely at this particular multi-electrode array, you'll see that there are a number of defined areas that we've created a device that sits within the multi-electrode array that perhaps can control where the cells are seated within this system. One of the other ways that we will interact with ourselves and some of the experiments that we've been doing is to use photostimulation. And so this is using a technique called optogenetics. Optogenetics is a form of synthetic biology where you can take a gene from an algae that creates a protein, and that protein reacts to light, and it opens a channel in the membrane of the cell, and that allows sodium and other ions to flux across the membrane, which can initiate an action potential, so generate that electrical activity in the neurons. And you can control that here with 470 nanometer light. There are different types of channel rhodopsins that have been designed and developed where we can use different wavelengths as well. And so this gives us a really precise way to stimulate particular subtypes of cells, because you can have these genetic constructs being expressed in different types of neurons. We can precisely time the light stimulation and the way that we measure from cells at the same time. So it gives us this very precise cell type specific manipulation of the cells, where we can perhaps use the light to inject information in the types of patterns of activity, and it also gives us that spatial as well as that temporal control of which cells get stimulated at which time. So in this particular video here, you can see that every time we pulse a 470 nanometer light into the cells, you can see that the cells light up. This is a fluorescent dye that is in a different wavelength, so that every time the cells are stimulated, you see the activity increase in those particular groups of neurons. So again, we can flash whole areas of cells with light, or we can be very specific into the exact areas, the exact neurons, depending on the light source, that get stimulated within a network. We've moved away from a lot of the dyes that we've used in the past, the fluorescent calcium imaging dyes, because they're detrimental to the cell's health over time, to using what's often described as genetically encoded calcium indicators. This allows us to do an all-optical approach where we can stimulate the cells using a particular wavelength of light, and then also record the activity of the cells using a different wavelength. This particular construct from this virus that we've used to express this Jacky in these neuronal cells is only being expressed in neurons. So this then again lets us record from different subtypes of cells, and we can use different colors, different fluorescent proteins to pick out the activities of different cell types. a benefit of using this system is that these particular cells are about 6 months old, and they've been expressing this particular genetically encoded calcium indicator for about 3 months. And so again, over long periods of time, we can look at the activities of our cells. Unfortunately, neurons don't stay still. They move around quite a lot. So then we can start to track cells in time in their movements as well as their activity. And so here you can see different pulses of the activity of the cells within these cultures. So light gives us a really high level of resolution in terms of stimulation, but also capturing the activity of the entire network of our neurons. We also want to be able to perhaps gain topological control, so architectural control of our neuronal networks. And so we can start to use things like microchannels to define how our neurons connect. We also know that neurons don't like particular features. They like soft materials. They don't like triangles. They don't like sharp edges. And so again, we can use topology to control their growth. And that can encourage modularity that we're enforcing using these structures. Gives us both long range control and maybe also local coupling of different networks. And so if we wanted to make a particular neuronal network, we could design a particular microchannel that could hold different subtypes of neurons and allow them to connect in ways that we could design into these systems. So in the typical design process, we may come up with very simple designs to more elaborate designs. And some of the technologies that we have here at Loughborough to make these devices can go down to the nanometer, micrometer resolution in terms of channel design, channel length, and some of the internal structures within these areas as well. And decide how the networks can connect within these areas. And if you look here in this image, you can see a very zoomed in connective area between these channels where you can start to see the neurons connecting in the different areas. So again, if we wanted to have different types of neurons in different regions, that we were able to use this design process, these micro or macro fluidic devices, to control where neurons actually sit within a network. And in terms of neuron connectivity, that we could start to maybe bias the connections between our neurons. So we can look at the internal geometries of those channels to very simple designs where we try to bias the likelihood of one population of neurons being able to migrate and extend neurons into another area. And here you can see some cells expressing M-cherry, so a red fluorescent protein here from a sea anemone, extending into an area of green fluorescing cells, so expressing green fluorescent protein. And what you start to see, hopefully, is that in this area you can see red cells. It probably shows the wrong colours for myself, I'm colourblind, but you can see, I can even see that red cells in this area amongst the green cells, but you don't see green cells in the red zone. So these green cells have not been able to migrate within this channel design into the red area. So again, start to bias the populations and the way that they connect to each other. And so we can start to bring these different technologies together. And so here you can see one of these designs on the multi-electrode array of one of these simple devices. So this is a PDMS, so it's a type of silicone device that's seated within this multi-electrode array. And in this video, you can start to see where those networks are connected and even the channels in between these regions of these networks. And you can see the activity of the cells spreading through those channels to these different connections. And so over time, we can model the connectivity of those cells. And when we stimulate the cells in different patterns in different areas, we can start to look over time and we can grow the cells on these chips for 6 months or more, how that activity develops within a culture. So shameless tag to one of the papers that our group have recently published is how to actually make these devices and how to attach them to the different types of microelectrode array that are on the market. There are a number of different designs. They have different types of surface, and so this paper was recently published to start to show people how to make these devices, but also how to seat them into the different types of multi-electrode array. So hopefully people can start to use these types of devices that we can manufacture and use them within multi-electrode arrays to design their neural networks and also start to link them to activity maps. And Jacky will touch on more of this data later on, and those connectivity maps of how these neurons connect to each other. So as I just come to the end of my talk and the areas that we're working in, hopefully lots of you are aware of the work on cortical organoids and other types of brain organoids and assembloids, where we can start to put together different organoids from different brain regions. These are organ-like three-dimensional cultures. In these particular cortical organoids, you can see they look a little bit like pineapple rings. These are cortical rosettes that are going on and developing to form the different cortical layers within this three-dimensional culture. Okay, these are free-floating structures that we maintain in the lab for periods of months or years to see how they develop over time. And then in the picture here on the other side is an image of one of these organoids that's been plated onto a two-dimensional surface, and these organoids have attached to that surface, and then they've started to spread neurons out from that organoid structure. And so again, by using organoids, by bringing these different technologies together, we're able to connect organoids of different areas, different subtypes, using different designs of channels, different channel designs, internal geometries, to see how these organoids connect and how they start to communicate across these structures. And as each of these organoids is oscillating, you can start to see the connectivity between them. And you can see the fluorescence as it travels through the channel. And you can start to see how one organoid can start to stimulate the other. And if we use optogenetics and drugs, as well as the electrical activation from the multi-electrode arrays, that at many levels we can control the activity of our neurons, as well as recording very high ratios using microscopy, using the multi-electrode arrays, all of the cells that are involved in that activity. And so again, a lot of information can be generated using these different types of assay. So I've given you a whistle-stop tour of some of the things that we're working on in NeuroSYNC and some of our previous work as well within the new chip project. And hopefully you see that we're combining a number of different technologies together using these stem cell-derived neurons, using both optogenetic inputs, the high density multi-electrode arrays to measure outputs, but also have inputs into the system. We're developing different ways to have some kind of architectural control over the neural networks. And by using these approaches, trying to study computation as it emerges within these human-derived neurons, so we can look at this process over time and use this as an inspiration for maybe next-generation computing paradigms, as well as maybe using them in themselves as biological computers. So as Natalia mentioned at the beginning, we have a neuromorphic computing center. It's a very multidisciplinary group. There are lots of us that work together in these different areas. And it'd be great, it's great to talk to different people from different disciplines, because the different techniques that we need to use to bring these kinds of projects together, it's very important to work across different disciplines. A number of different companies that we work with, particularly on some of the work in the biology labs that we're doing together with BitBio at the moment, in working on how we make the different neuronal subtypes that we can bring together in these cultures. We'll have some time for questions at the end, but if you'd like to contact me separately, then you can get me at my email address at Loughborough University or via Natalia in the Neuromorphic Centre at Aston University. Thank you very much for your time, and I'll now hand over to Jacky.
Hey, thank you, Eric. So can you all see my screen? Yeah. Okay. Okay, so hello everyone, I'm Jacky. So lecturer in applied mathematics at Aston. So while Eric already shared a lot about the biological side of the biological neuronal networks, so I'm going to talk about more about the math, and from a more mathematical point of view on understanding the biological neuronal networks. So we will first talk about the motivation of our previous project, the NeuroChip project, and some of the work that we have done. Then we talk about some challenges we are facing and the methods and approach we employed in order to tackle all these. So as Eric mentioned, the current machine learning and AI tools are consuming a lot of energy. So growing and keep increasing over time. And therefore, it is a need for us to explore new computing technology, and biological computing, as Eric suggests, has a very strong potential. So we want to create an AI chip that using human cortical cells And how to do that? And in particular, we will put the cells and the culture that Eric mentioned and grown onto the MEA chip, so that we can see the activities and allowing us to do stimulation for training and also input-output testing, as you have seen in the previous presentation. So based on all these, so we have created a neurochip prototype, which aiming to do a very simple classification task on the pattern we are stimulating. So so first of all, the culture is grown and put on the MEA. Then we applied our training protocol, mainly based on the LTP stimulations, long-term potentiation, trying to induce the connections between the region, that is the yellow one with the pattern 9, with the output area on the right-hand side in red. And while we are trying to connect the pattern area 0, the green one, with the output area in blue on the left-hand side. So after we do the training, we will apply stimulations on the corresponding area, like we stimulate on the yellow one, electric stimulation, in the yellow region, and we will test the corresponding activities on different regions. And we will also stimulate 0 and compare the effect. So on the right-hand side, you can see, so before the training, this is the baseline activity. And while after the training, we can see that the response of the right-hand side, the red curves, responding to the stimulation of pattern 9 is much stronger on the right-hand side than the left-hand side. So it seems the neurochip device prototype is working. So while we can do some simple tasks using these first prototype, so however, what is happening inside is still a black box to us. So in order to understand what is happening inside to give us insights to create a reliable computing device, so we must understand what is happening within the chip, within the culture. So there are 3 different things that we are particularly interested in at the moment. So the first thing is the network functionality. So from the culture, they are doing their things. They are spiking and propagating signals across the networks. So what we want to know is if we can identify the effective structures, in particular, which neurons is affecting another neurons throughout the spiking activities. So this is something that we want to know. Another thing is about the emergence of content, whether a particular stimulations will affect the spatial entropy of the culture, or how the entropy will change over time as the culture grows. And the third thing that we are interested in is the development of the culture over time. So we have, in each day, we have different recordings, but they are difficult to tell what are the differences. And we want to understand it in a more intuitive way. So the first thing I would like to talk about is the network inference that we are doing. So first of all, inferring the structures from these spiking activities is still an open question in neuroscience, which is very challenging. And yet it is a very important thing to know, because it is the key to help us to understand how plasticity and the learning dynamics of these cortical culture works. And by extracting the current network structures, and can help us to understand what is the current state of the culture, and then we can apply appropriate stimulation strategies to do the training and let the culture to perform tasks. On top of that, it also gives us some insight on the functionality of the cortical domains, as well as giving us some insight on neurological disorders. For example, if you can see there is some significant difference between cultures with disease and the healthy one, that is something very interesting. So what we are aiming at is to find a probabilistic method that allowing us to infer several things. So the first thing is about the neuronal cell types, whether a cell is excitatory or inhibitory cells, as Eric mentioned. So excitatory cells basically is neurons that will send, when they spike, they will send signals that stimulate their neighboring neurons to spike. and while inhibitory neurons prevent their neighbors from spiking when it spikes. So another thing that we would like to know is the existence of link. So current methods, most of them are assuming the graph is fully connected, while this is not something realistic in biological culture. So we want to identify whether a connection between neurons exists or not. And the third and fourth things are we want to have a probabilistic method allowing us to know the interaction within the cultures between multiple neurons and allowing us to do neuronal activity predictions. So to do that, we are trying to map the binary neuronal activities onto a model called the kinetic Ising model, which is an non-equilibrium statistical physics framework. So if we look at the neuronal activities as a binary state, or either they are firing or silent at a time point t, then we can define a transitional probability, which the state of neuron Si at time point t is depending on the background activity term Hi and the effective synaptic strings Jij coming from a neuron J in the previous time point t. So while this is just normal physical model, it's not enough to make things realistic and align with biological setup. We also introduce latent variables, set J, which is the cell types of the neuron, as well as the phi ij, which is the existence of link from J to I. So the goal of our model is to trying to infer Jij, Hi, Zj, and phi ij all together simply from the firing activities. And to do that, we employed a method called generalized maximum likelihood, or people also call it evidence approximation. Which is a Bayesian method, and we employ it through a two-layered expectation maximization DM framework. So since it is very difficult to check the ground truth topology in real biological dish. So as you can see, the network is complex and is fairly challenging to identify the cell types. So the first thing we do is to simulate a neuronal network and check our results with emulator data. So we simulate a neuronal network with biological ratio of 80% of excitatory cell and 20% of inhibitory cells that is similar to the human brain. And then we model them on a stripes pattern substrate that you have different stripes here. So it forces the high connectivity within each stripes while lower the connectivity across the stripes. So we simulate the activity and use the activity data to infer the structures, and our algorithm performance is actually very well. So you can see that the inferred structures actually capture the strong clustering effect created by the stripe substrate. So each of these squares corresponding to one stripes. And on the other hand, you can also see that our algorithm has a very high predictability rate of identifying different cell types of neurons. So in particular, you can see around 87% of overall accuracy and 96% for excitatory and 64% for inhibitory neurons. And also, it is working quite well, very well in terms of identifying the effective links over the network. So having validated the method in silico, we move on to work on real primary cultures data. So we used experimental recordings of spontaneous activities from a wet cortical neuronal culture grown on a patterned PDMS substrate, so which also have different stripes and suppressing the activities, connectivity across stripes. So they are recorded on 17 DIV, day in vitros. And what we are trying to do here is to use the activity data and again to infer the structures. So as you can see in the connectivity matrix here, so our methods also, just like the simulation result, the inferred structures show a strong clustering effect forced by the physical PDMS substrates. And when we plot them as a network format, you can see they are densely connected within stripes and only a very few connection across. So this is the pictures of the calcium imaging. So to further validate our work, and we use the inferred parameters, the J and the H and the Z and the Phi, and to generate artificial neuronal activities using multicolor simulations, and to compare the predicted equal time and also the delayed time covariance of the neuronal activities against the true experimental data. So the result showed a good alignment, which is the dotted line here. So better aligned. So to this dotted line, the better the results, and showing that our methods work also very well on real and noisy biological data as well. So next, the next thing we are looking at is to understand the neuronal culture using a visual informatics approach. So when we look at the neuronal activities as a system of n neurons, and each of these neurons have two states, so firing or non-firing. So if you have a culture of n neurons, there will be 2 to the power n of total possible firing patterns, which is an extremely high dimensional space, and it is nearly impossible for us to gain any insights by just looking into the standard raster plot. So to solve this problem, we are trying to use visual informatics technique to trying to embed all these high dimensional data point. High dimensional data point onto the lower to the two-dimensional representations, and hopefully from these lower dimensional plot, and can help us to understand and analyze the data to get insights in terms of culture development and also the culture response to stimulation. So to understand how exactly a culture responds to stimulation, we track a primary mouse culture grow on an MEA device over several days, and before day 14, we only record their spontaneous activity, while after day 14, we will introduce target stimulation protocol and record the post-stimulation activity. So down here is the some activity record on day 14. And right-hand side are the spiking activities, and left-hand side are the neuronal traits. So to visualize the activity data over different days, so we apply a method called minimum distortion and banding, which is MDE, which is a dimensionality reduction technique that trying to retain the global structures of the data on the original dimensional space, and also retain their relative distance, which is much better than TSNE, because TSNE only focus on the pairwise distance between data points. And as you can see in here, the right-hand side are the embedded results. And you can see the activities at different dates are plotted in different colors, and they are forming into distinct clusters. So suggesting that the behavior of the culture are grouped into one cluster on the same day. And you can also see that when they over time, you can see a little trend going on here to here. So suggesting that we can show the culture development over time. And also, interestingly, you can see in here that this little circle. So these stars are the clusters of the stimulated activities. So this is the activity recorded while we apply the stimulation to them. So we can see that all these activities being stimulated are formed into clusters, suggesting that we are guiding the culture in a way such that the activities will have a similar behavior when we stimulate. Okay, and another thing that we have worked on is on the information content, understanding it from a spatial entropy point of view. So because of time, I'm only talk about very briefly. And the general idea is we want to see how the spatial entropy of a system change when we apply chemical or electrical stimulations. So on the left-hand side, we have an activities that is recorded and applied drug to suppress the activities. So we can see that when we apply the drug stimulations, which suppress the activity, the entropy of the system will drop, suggesting they are performing more organized. And while we wash off the chemical, and the entropy jumps up, so suggesting they are less organized and more random. And on the other hand, on the right-hand side, this is something about electric stimulations. So here are the activities over day recorded, and we measure the entropy over time. So you can see that when they are young, the entropy is very low. And when they are mature, the entropy is much higher because they are more vibrant, more active. And on the days that we apply stimulation, we can see that the entropy actually dropped. So suggesting that that the culture is responding to the stimulation we applied. So this method can help us to identify a strong plasticity effect when we explore the values of the entropies. So here are the summaries of what the things that we have covered so far. The first thing is we work on network inference, so identifying the cell types, the connections, and the strings, verified using both computer emulator data and also realistic data. And we also understand the culture in, using visual informatics to see if data in different days have their own characteristics that can form into a distinct clusters. And the final things that we have worked on is to understand things from a information content, specifically entropy point of view. So in the future, there are a few things that we are aiming to develop. The first thing is to try to identify the optical training protocol that make our new chip prototype and better. And we want to use the methodologies we developed to understand plasticities and how brains work better. And also, same as Eric suggested, and trying to extend our work to a 3D organoid culture instead of just a 2D culture. And all these developments can help us to explore new computing algorithms, not only on biological, but also in computer or in other neuromorphic computing areas. And however, these things are quite challenging because of different aspects. First of all, because the lifespan of the culture, they will die somehow. And also, it takes a very, very long time to grow, so which is resources intensive. And the scalability is also one of the problems that we have to address in the future. So that's the end. So if you have any questions, please feel free to ask Eric and I.
Thank you, Jacky. Thank you, Eric, for wonderful talks. Do we have any questions? You feel free to pop it in the chat or just raise your hands. It seems that we don't have any questions. I don't know if it's good or bad. But anyway, I think if you will have it afterwards, just feel free to email us. We are happy to answer. Again, as we mentioned, we are very open for collaboration. So anytime, just fill in the form at the website or just send us an email. Thank you, everyone, for coming today. Hope you enjoyed it. And we hope to see you next time. Thank you. No, we have some questions. That's great. So, how do you plan to create these algorithms? Feel challenging to pass from the emergence to digital?
So, do you mean the algorithms for doing the computing task, or the algorithm for doing the inference of structures?
Yeah, sorry, it's me. I'm gonna talk. I cannot turn on my camera. I'm in the street. I mean, so neurons are kind of both digital and analogical. So how do you plan to build all these digital algorithms while extracting all this data? And I mean, like trying to create, I don't know, forests, or I don't know, things like that.
I'm not sure.
So
I
mean,
completely
you
follow
extract
directions.
all the information. We've seen all the information from the multi-electrode arrays, all the spikes that you can get. How do you transfer all this information into a algorithm that may be used, I don't know, for AI?
I see. So, well, because this is a very, very early stage. So what we are trying to do is to do a very, very simple classification task that basically relying on the plasticities and the connectivity between cultures in different areas. So we do not assume, and we do not know how exactly these algorithms we should design in order to do the computing. But this is the first step we are applying. But you can imagine something like a reservoir computing. Then you have over a network. Then neurons are connecting to each other in a chaotic way, and information will be propagating over the network. And this is one of the directions we are exploring too.
Okay, thank you so much.
Thank you.
There's another
Yes,
question. I
David?
wasn't quite sure what it means by what is the fan in that can be achieved. I wasn't quite sure what that means. If this the person who wrote the question is still available.
Okay, that was for me. I don't know whether you can hear me.
Yeah.
I was just wondering, because one of the advantages in the natural brain tissue, so it's a very high connectivity. So the visual cortex, so fan-in for a single neuron from 10,000 other neurons. So it was mentioned that you have 100,000 neurons in your cultures. I was wondering, so what connectivity you can achieve? How many inputs to a single neuron?
Yeah, so these are some of the different areas that we're exploring in NeuroSync. So that is in the entire 3.8 by 3.8 millimeter MEA, but using the devices, we can use much fewer neurons and also control how many potentially connecting or the weighting, perhaps by the size of the channel, the design of the channel, into those different areas. So how you can go from an area that could be larger, where we, as Jacky showed in the image there, that was quite a large rectangle with quite a lot of neurons in that area, could be funneled down to much smaller areas, more defined areas. So different ways of controlling that using the architecture of the design of the devices, but also chemistries as well, in the way that the neurons are attached and encouraged to grow into different areas. So within those designs, having different input and output areas that we can put into those networks is something that we're also doing at the moment.
But those two-dimensional channels, they will very much limit that fan-in. So I was wondering, so what is the highest number you can reliably get in inputs to a single neuron?
So on the MEAs, we are restricted in that particular MEA by the size. Some of the other multi-electrode arrays from different companies have more electrodes, a much bigger area to grow neurons in. For some applications, using microscopy and optical approaches may give us a much larger field of view with the constraints, I guess, of the size of the lens that we're using to look at the cells. But we could also have multiple chains of MEAs linked together if some companies allow you to design your own multi-electrode arrays, and so that may give us much better control rather than off-the-shelf systems.
Maybe it was still a misunderstanding. I'm interested in how many inputs you get from other neurons into one neuron in your culture. So not the interface from the outside, but within your network. So how many other neurons can connect to a single neuron?
So it is not a sorry. So this is not a computer design structure. So it is not us to assign how many neurons is connecting to one neuron. It's them will have a self-organization, and they will form axons and connecting to the not form axons. The axons will grow and connecting to the dendrites, and in a spontaneous way.
Yeah, so, I understand this, but in a brain, one of the amazing things is that you have 10,000 inputs to a single neuron. So I was wondering, what is your sort of input fan-in to one neuron?
I guess at the moment we don't know how many, unless we use imaging to define how many neurons are connecting to a single one. It's often that we can try to bias in different areas how many neurons are there, and then allow them to connect in the way, because we have to put them in as progenitors into the systems and then let them develop. And so we can bias that initial plating into particular areas with different amounts of neurons, and using the channels to reduce and control the number of neurons that can go into that. I guess we don't have a definite answer as to how many go into each individual neuron at the moment.
Okay, thank you. Thanks very much.
Thank you. We have another question. We still have 5 minutes, so probably we can take one more.
So Jacky, I don't know if you can see that question about the noughts and zeros on organoids and how they're represented. So it's an
Yes.
interesting question about how they might represent that. It's interesting, and using the MEAs, it may not be ideal for some of the work on organoids that we don't necessarily, perhaps using some of Jacky's methods, infer the connectivity within the organoids and how certain inputs might be represented. A lot of the times with the MEAs, we're measuring parts of the organoid, and we may be able to infer from the activity that you can record what might be happening inside the organoid depending on the size. So some of these methods might not be ideal to see in the detail the way that I guess those particular digits may be represented.
And I see David also has a question.
David had a question, yeah.
No, I don't have questions, I have comments. So the first one about the learning methods. So the idea is not to replicate existing training methods from computer science on biology. We're trying to exploit the fact that these training methods have been developed naturally. So we're simply trying to gain correlations between inputs and outputs through joint stimulation. If you want, it's similar to the Hebb rule, but obviously it has worked for millions of years. So that's what we want to emulate.
Thank you. Thank you. Karanpreet, you have another one?
Yes, sorry, I know I'm a bit late, but thank you for the wonderful talk, Eric and Hofe. I just had, so you talked about, we are talking about learning algorithms. You talked about kinetic Ising model. I was just wondering, I mean, definitely your work shows that, we have good agreement between in silico and in vitro, but do you think you might be losing some state of every individual neuron because you are just mapping it to a binary
Yes.
activatory versus non-activatory? Because I assume that in the networks there might be some transitionary states as well. And is there, and this is my first question, and second question is that, is there a certain amount of time that it takes for the network to settle after you give it some activation? Does it take some time to settle, or is it like, an instantaneous kind of a process?
Yeah,
Okay.
I think so part of the first question, I guess, in some of the way that the multi-electrode array may record information, we should be really careful with how we threshold spike detection in those. But I do think that imaging gives us a lot more flexibility in looking at events that may not lead to a spike that might be, say, recorded by a multi-electrode array, that we can see activity in really defined areas of a neuron as well that may not reach the threshold to cause a spike. So I think the imaging gives us a really sensitive readout of things that we may miss by having a very binary recording. Jacky, sorry.
Yeah. So definitely we are, same as Eric mentioned, we are definitely losing some of the information. So for example, like when we are recording the activities, they are on the electrode. So on a single electrode, there might be one or two more or even three neurons on there. So definitely the recording will lose some of the information. Not only that, when we employ a kinetic Ising, we assumed a causation only take from t-1 to t, so only one step. So a longer range correlation is not taken into account, and also something biological is also not taken into account, like a neurotransmitter reservoir reserve that will facilitate how a neuron spike. There are several things that we are missing, but the idea is we want to find a simple a model that is simple enough and effective enough to understand what is going on inside. So when we take more and more consideration into it and put more and more parameters into it, of course we can not definitely, but we will have a higher potential to get a better inference, but it will also increase the computational complexity of your algorithm, and sometimes you are just gaining something less important. So this is something that we think is in a right balance. So a model that is simple but is quite effective to explain what is happening.
Perfect. Yeah, thank you. Yeah, because especially if the second question was more about if the model takes some time to rest after the nudge, so I could see it kind of like an equilibrium propagation model, or either some sort of a vector field model that you're nudging it. It's a sleep wake cycle, so it activates and deactivates, and maybe that can be one of the ways that we can think about training these. But anyways, thank you for a fascinating talk. Yeah.
Thank you very much. Thank you for all the questions. Thank you again for coming. Thank you for wonderful talks, and see you next time.
Thank you.
Thank you.
Thank you.